Okay, class. Let us begin. So, in today's class, we people are gonna be looking on toward the classification thing, and over the many years, I have realized that the students of IGCSE, GCE, O levels, or whether they are from the Excel stream, so they suffer with this topic. So, if you guys can clearly look on toward the title, so this is the classification thing with us, and we have to be very much clear that what this classification actually is. You see, if we go a few years back, so the human beings they were not bothered about the living organism, so they had got no idea of whether we are supposed to be placing them in some specific groups or in some specific divisions until as the advancement came into the biology with the introduction of the microscopes and the X-rays and a lot of the stuff like the gel electrophoresis techniques, like we were advancing in terms of the diagnostic things. So then the scientists they said that okay, fine, let us just try to be classifying the living organisms because every passing day when they started interested about looking on toward the outer features of the organism, so they said that why not to be placing them in some specific groups. So why they were doing it? With an intention to be making the studies of that living organisms easier because whenever we are coming across some new organism randomly, so we have to be clear about them that whether they would be plants, whether they would be animals or the fungus or the protoctus, or whether we should be placing them into the bacteria. So thanks partly to this scientist that we have, he was Linnaeus. And this scientist, Linnaeus, he started to be placing the living organisms into the groups. And the idea is very simple that we have to be making the study of the organisms easier for us. Like if I were to quote you some example from your daily lives, if I were to say, that you have got in your home some kind of a home library, and in that home library you have got some of 100 books in your collection, and these 100 books they were messed up and you don't know that what topic they were discussing or what idea they were going to give on to you. And on one random day, you decided that why not? I try to be categorizing these books into the unique categories. So when you opened up the book and then you started to look on to some of their initial pages, so you said that okay, these are the Islamic books, these are the science books, and among the science books, okay, these are the biology, the chemistry, and the physics books we have got. So when you will try to be putting these books into the categories, so next time when your father is gonna go into the market and he is gonna bring some new books for you, and when you people are gonna be opening it up, then depending upon the title that book is gonna have or the context that book is gonna have, so it would be easier for you to be placing these books into the specific category. So ultimately the categorization would become easier. So this is what the Linnaeus was, the scientist or the biologist that basically started to be doing this. Now what he actually did, that he started to be starting from the very major kind of or very basic kind of a category, generalized kind of the category, and he formed the domains. Like we have got three of the total domains with us. Among those domains, we would have the bacteria, we would have the animalia, and then there was Archie with us, but that is not discussed at your level. Then he started to further subcategorize them, and we're gonna see in a while that what are the kingdoms, the phylum, the classes, and ultimately we would end up being the species. So this is the approach which the Linnaeus basically followed in order to be classifying the living organisms. But for our course, we people have to be very much clear about the classification thing that we have got in terms of putting the organism into the vertebrates and into the invertebrates thing that we have. Like a lot of work was done by the Linnaeus, but here we people are gonna be looking on toward the basis of the classification. So you just need to have an approach that for the animals thing we have got the vertebrates and we have got the invertebrates, and this word vertebrate is not something new because we know all those organisms that are gonna have the spinal cord in them, so they would be called as vertebrates. And among the vertebrates, the major classes that you people need to have an idea of, so we have the mammals, we have the birds, we have the reptiles, we have the amphibians, and finally we people have got the fish with us. On the other way around, we have got the invertebrates with us, and among the invertebrates, as the word itself indicate, that they gonna lack the spinal cord in them. So scientists, what they did was that okay, fine, if they were coming across invertebrates, and in those invertebrates, if they were to see that their body had got some segments in them, and in each of the segment, if they were seeing the pair of the legs. So such invertebrates, they call them as arthropods. So for your course, you people need to know that among the invertebrates, we have the arthropods, where the body of the living organism, it would be made up of multiple segments. Like if you guys could see over here, so we have got multiple segments in them. And among the segments, you see that we have got the paired legs in them. So such are the arthropods with us. And among them, the people need to have an idea of the insects, the myriapods that we have, the crustaceans we have got, and the arachnids we have. How come this be possible that we be skipping the plants? So among the plants, you guys need to have an idea of the flowering and non-flowering plants, we say. Now, as far as the non-flowering plants are concerned, so they would include the ferns and the mosses, but our more discussion would be regarding the flowering plants. Now, among the flowering plants, we further have got two major kind of the subdivisions in them, like we're gonna categorize them into the monocotyledonous plants and then into the dicotyledonous plants. Now the situation is that when the Linnaeus was doing the classification, so he basically realized that everywhere in the world, the living organism, they are given a different name, and that was making confusion. Like if I were to talk about myself, so everywhere in the world, the cat has got some different names. Like in my country where I'm residing, so we use an Urdu word for it, and that is Billy. So this was creating a lot of a language barrier in terms of identifying the species. So what Linnaeus actually did, that he started to give the living organisms Latin names. Like if I were to talk about myself or yourself, so generally we call ourselves as the humans. Now human being is something very evident, like we are being discussed, but this would be there would be some other word for describing a human being. But when the Linnaeus he gave the scientific name to the human beings, so he said that they are Homo sapiens. So Homo would be relating to the genus, and the sapiens would be relating to the species. And we're gonna see in our next lecture that how come we're gonna be writing the scientific name of the organism, and that name, scientific name that we give to the living organism, now that is around or revolves around the binomial system, that it is going to be comprising of the genus and this would be comprising of the species. Now the situation is that how come I am able to classify a living organism? Like for instance, like I went to a forest somewhere, and in that forest I came across some living organism, and when I started to be looking on that living organism, I basically realized that its body is covered with the hairs. It has got some external ears on the body, but I'm not sure whether I should be putting it into the mammals or something else. So you see that we must know that what are those bases for the classification thing. Like if you come across anything new in your life, so how you gonna classify it? Like whether you're gonna be putting it into the mammals, into the reptiles, into the amphibians, or into the myriapods or the situation like such as I have indicated above. So first, guys, we people are gonna be looking onto their outer features. Like as in case of the humans, the outer feature means our body is covered with the hairs. The females or they're gonna be feeding their young ones with the help of the mammary glands. Okay, so presence of fur. So these are the outer features. But in some situations, these outer features they are misleading. So the other criteria which we have got to be more clear about it into what groups of the vertebrates or invertebrates we're gonna be putting them up, then the second approach is that I'm gonna look on toward the sequence of the nitrogenous bases, and that sequence of the nitrogenous bases is gonna decide that whether the living organism is really the human being or it is something else. And if we are even not convinced, then the third approach that we have got to be classifying the living organism into some specific category. So we people are gonna be looking on toward the sequence of the amino acids that we are gonna have in their proteins. So if I were to summarize, I'm going to look on toward the outer feature of the living organisms. Then I'm going to look on toward their sequence of the nitrogenous bases or the sequence of the nucleotides that we people are going to have in their genes or the sequence of the amino acid that we people would have in their proteins. Now, what do we mean by all of this? We people are going to be looking in a while. Now you see that the first approach to be classifying the living organisms on the basis of the outer feature which they have. So this is not something difficult. But here we must clear our idea that if I were to classify the living organisms on the basis of the nitrogenous bases or the gene sequence type which they have in them, then how that could be done. So, guys, the situation is that at some point or the other, you people may have come across the cell of the animals, and in those cells of the animals, now here you see we have got this outer boundary in them that is the cell membrane. And here you see this is a part of the protoplasm that we have, and it would be regarded as the cytoplasm. And here we have got the nucleus. In the nucleus, we have got the chromosomes. If I were to be showing you the chromosomes, so here you see, guys, we have got a chromosome here, and in the chromosome, we have got a narrow constriction at the center, and that narrow constriction would be regarded as a centromere. And to each of the chromosome, like in the duplicated form, we gonna be seeing two chromatids in them. Like this arm of the chromosome is one chromatid. And this second arm of the chromosome that we have got, this is the second chromatid with us. Now, this is something not important for us. The other thing that is important for us is that within each of the chromatid we have got here is this molecule, and this is the DNA molecule with us. So if I were to understand the classification on the basis of the sequence of the nitrogenous bases in the DNA molecule, then I must know that what this DNA actually is designed, and that would help me to be classifying the living organism. But before we proceed, now here are two of the keywords that you guys have to be very much clear. Among the first, we have got the word polymer. Now, what polymer is, suppose if you people were sitting in the room and over there you have got the wall, so wall is a polymer, and that wall is made up of the small repeating units in it, and those small repeating units, they are the monomer. And in this situation, this would be the brick with us. Okay. So this means that if I were to say that this DNA molecule is a polymer, then I need to get an idea of its small repeating unit, which is a monomer with us. When I would get an idea of the monomer, then ultimately for me the approach of the classification in terms of the sequence of the nucleotides or the nitrogenous bases would become easier then. So before I go with making you understand the structure of the small repeating unit of the DNA, for the chromosome you see that it has got in itself this one centromere, and then this has got total of two chromatids in it. Okay, so here we have got two chromatids, one centromere, and in each of the chromatid we have got two DNA molecule. And overall, for one DNA molecule we have got two strands, and overall, in one duplicated chromosome, we people are going to have four polynucleotides or four strands. So let us just now try to see that what thing we have got to be understanding in terms of the DNA molecule. So if you guys see here, so this DNA molecule is a double-stranded molecule. And technically speaking, one strand of the DNA, this would be referred to as a polynucleotide, and the word polynucleotide is indicating that I have to take nucleotide. I am gonna join them again and again, and then ultimately we will make one polynucleotide or one strand of the DNA. So as I have said earlier, that this DNA molecule that we have got with us, so this is a large molecule. Like you guys have to see that this is a polymer, and this is possessing some small repeating units, and they would be referred to as the monomers. And the monomer that we have got in the DNA molecule, so that is the nucleotide with us. So you see that if I were to say that nucleotide is the small repeating unit of the DNA, then you must know that what other components we have got within a nucleotide. So we have got total of three components in one nucleotide. And among those three components, the first thing that people are going to have in it, so it is going to be the nitrogenous base. And why the word nitrogenous base? Because they have got nitrogen element in it. The other thing which I'm gonna have in a nucleotide would be a sugar. And the third thing which I'm gonna have in a nucleotide of a DNA, so that is gonna be a group, and it would be phosphate group. Like in chemistry, you may have learned about the word phosphorus, isn't it? So from that, you see that this phosphate group is phosphorus based. Now, the situation here would be that you guys need to have an idea of that what would be the symbolic representation of a sugar for this one nucleotide I'm talking about. So the symbol of this goes like this, like you have to be making a block. the phosphate is gonna look like this. Like you have to be putting a circle and then you are gonna be writing the P into it. So this would be the phosphate group. And if I were to show the nitrogenous bases, so whenever you have to be showing the nitrogenous bases, you must remember is that in the DNA molecule, the total nitrogenous bases are four. Like it would be A for adenine, G for guanine, C for cytosine, and T for thymine. So, if someone wants to ask me that you make for me the nucleotide, adenine containing nucleotide, so I'm supposed to be making a sugar, I'm supposed to be making the phosphate, and then you see, I'm gonna be writing A here. So, this is adenine containing nucleotide. Now, I'm gonna join it with the, you could say, thymine nucleotide. So, here I would have the sugar, then I'm gonna have the phosphate group, and then I'm going to write T here. So you see that this one repeating unit that we have got for the DNA, this is one nucleotide, this is other nucleotide. So when the nucleotide would be joined with one another, so we would make a strand out of it. Okay, what did I say? Strand out of it. So this is one DNA strand. And likewise, there would be the second strand in which the nucleotide would be attached. And by that, we people are gonna be making the second, you could say DNA strand or the polynucleotide. And these polynucleotides, when they are gonna be joined together, so they would make one DNA molecule. So the living organism which I just encountered in the world, and I was not sure to be placing them into the mammals or the reptiles. So I am gonna take the DNA out of them, like the blood out of them or some hair follicle out of them, and then I'm gonna take the DNA, and in the DNA I would look on toward the sequence of the nitrogenous bases. If this is close to our sequence of the nucleotide that we have got in the cells of and in the DNA of our cells, so we would say that okay, fine, this is a human being. But if this sequence that I have written as such, like ATGC or something situation like that, if this is not similar to our sequence of the nitrogenous bases of the nucleotide, so we would say, okay, fine, this is not a human being. So in the classification, the first approach would be to be looking on toward the outer feature. If you are confused and you here you have to come on toward the DNA, and over there in the DNA you would look on toward the sequence of nucleotides or toward the sequence of the nitrogenous bases, and then you have to compare it with the known sequences for the humans, maybe for the plants, maybe for the funguses, and then you're gonna be categorizing it as such. So if I were to summarize this, so I have to have the nucleotide, I'm gonna join them together. So we people are gonna be making a polynucleotides out of them, which is one strand, and these two polynucleotide, when they would wind around each other, so we people are gonna make the DNA out of it. And in the DNA, we have to be bothered about the sequence of the nitrogenous bases. Now, the third approach is that we use to be classifying the living organism. So this is protein with us. Now, you guys must keep it in your mind as that this polymer that we have, so this again is a large molecule. I said protein is a large molecule. This is a polymer. So if this is a polymer, then I have to be understanding as that what repeating unit I have got in these proteins. Like in case of the DNA, this was nucleotide. And in case of the protein, this is amino acid with me. At some point, you people may have eaten up the chicken piece, okay, like some leg piece or a situation like this. So here you see what you eat at your KFC. Now this is overall the protein molecule. And while trying to eat it up, so you may have split it up. And when you would split it up, then you would have come across some strands. Now, this is for your understanding. The strands, they are like highly microscopic, but you must know that these strands that we split out of the protein or out of the chicken piece, so this is gonna be the polypeptide chain. And for sure, this polypeptide chain that we have got, it would have some small units into it, and these small units, they are gonna be the amino acids we say. So when we have these amino acids, they are gonna combine with one another. We will make polypeptide chains out of it, and these polypeptide chains, when combined together, so they would be making protein out of it. Now, what the situation is that any of the living organism that you come across in the world, you people need to be taking the proteins out of it. Now, where can we get those proteins out? Like in our body, in the human's body, we have the red blood cells, and in those red blood cells, we have a protein which is a hemoglobin protein. The antibody is a protein. Likewise, the hair that you have got, so they are made up of a keratin protein. Okay, so guys, the overall scenario is the protein is a polymer of small repeating unit. These are the amino acids. If I were to take the amino acid, if I were to link them together, So we would make polypeptide chains out of it, and these polypeptide chains they would make protein out of it. So if I were to just give you a situation, to present you a situation, if I say that I cut my fingernail, and in that fingernail I have got a protein, and that protein is a keratin protein, and in that keratin protein, the sequence of the amino acid it goes like these are some of the name of the amino acids that I have written. You're not required to be learning them, but to be getting an approach. So I have glycine, and leucine amino. Like there would be a large variety of these amino acids, but I'm quoting you the reference with respect to the three amino acids I have given here. So this is the sequence of amino acids that we have got in the keratin protein that I have got from the fingernail of the human being. Now, if I were to go in the world and I would come across an animal, the outer features were not able to convince me that whether this is a human being or not, then I would look on toward the sequence of their nitrogenous bases, and the other approach would be that I would cut the fingernail of that organism and I would have the keratin protein. And in that keratin protein, if I were to see the same sequence of the amino acids as we have got for the keratin that I have got from the human fingernail, so we would say that, okay, fine, this is a human being, so we should be placing it into the mammals. So, guys, this is the basic approach to the classification. In our next class, we people are gonna be looking on to the situation that how come the leniency basically worked out the domains, the kingdom, the phylum, and the situations like this, and we would see on toward the basic characteristics of the vertebrates and invertebrates. So see you in the next class.